Hello everyone, myself Dr. Parth Goswami, and today I am going to teach you about pathogenesis of glomerular injury. We will try to understand why glomerular injury occurs. So we will learn the pathogenesis behind the development of nephritic syndrome and nephrotic syndrome. So let's begin with glomerulopathy. So friends, basically there are two types of pathogenesis of glomerular injury. It could be because of two variety. It could be immune mediated glomerular injury, or there could be possibility of non-immune mediated glomerular injury, among which this immune mediated glomerular injury is the most common variety causing the glomerular injury. So let's begin with immune mediated glomerular injury. Basically there are four types of immune mediated glomerular injury. We can divide the pathogenesis of immune mediated glomerular injury into four variety. One is antibody mediated glomerular injury, second one is cell mediated injury, third one is alternate complement pathway system mediated injury, and fourth one is secondary certain pathogenetic mechanisms. So let's understand each mechanism in the detail one by one. So first of all, the most common mechanism leads to development of glomerular injury is a antibody mediated glomerular injury. It is a most common variety. Now here basically certain substance act as a antigen, and there will be formation of antibody against this antigen. That's why it is antibody mediated glomerular injury. And this formed antibody, this particular antibody will bind with the antigen and they will form the immune complex, right? So here there is a formation of immune complex. Now this immune complex antigen antibody formation can occur in the circulation, or it could be in situ immune complex formation in the glomerulus. So basically immune complex will form. Now if we talk about which are the antigen that leads to antibody formation, so antigen could be uh, you know present within the glomerulus, or it is outside the glomerulus. If it is outside the glomerulus, then immune complex will form in the blood circulation, right? So in the blood, substance that act as antigens are exogenous or endogenous antigen. The example of exogenous antigens are infectious products, certain infection like that of hepatitis, streptococcus, HIV, etc. Like, right? And endogenous antigen means the antigen are present within the body, not outside. Exogenous means outside the body, right? From the outside body it will enter into body, but endogenous means within body certain substance act as antigen. For example, DNA. If antibody forms against DNA, then classical example is systemic lupus erythematosus, or antibody form against the tumor antigen. So here the antigen present in outside the glomerulus and the antibody will form against it, which will form the immune complex in the blood circulation, and this circulating immune complex will ultimately get deposited in the glomerulus and it will lead to glomerulus injury. The another mode of immune complex deposition is in situ immune complex formation. Here the antigen are present within the glomerulus, right? It is a fixed antigen in the glomerulus, or antigen could be planted in the glomerulus from outside the glomerulus. Understand, from outside the glomerulus it can planted in the glomerulus and act as a antigen. So which are the fixed tissue antigen present in the glomerulus? So the classical example against which the antibodies form is a glomerular basement membrane. It contains the type 4 collagen and particularly NCI domain, against which antibody will form and it will lead to glomerular injury, which is known by the name Goodpasture syndrome. Antibody can also form against the membranous antigen, particularly glomerular basement membrane, visceral epithelial cell antigen, right? In the podocyte, there is a presence of PLA2R antigen, against which antibody can form the immune complex and mediate the injury, which is known by the name membranous glomerulopathy, which is a most important MCQ. or the mesangial antigen. Antibody can form against the mesangial cells as well. So these are the example of fixed tissue antigen present in the glomerulus. Now, which are the planted antigen? So planted antigen could be exogenous, like that of infectious agent or drugs which will plant in the glomerulus, or it could be endogenous DNA, nuclear protein, immunoglobulin, like that of IgA, etc. Understand? So basically, antibody mediated glomerular in the antibody mediated glomerular injury, there is a formation of antibody against the antigen. They will form the immune complex, and immune complex will mediate the glomerular injury. We will understand how. So most of the glomerulonephritis is an immune complex mediated glomerular injury. Here the immune complex will form in the blood circulation or in the glomerulus. If it is formed in the blood circulation, then it will get ultimately trapped in the glomerulus, right? And this deposited immune complex in the glomerulus will mediate a type 3 hypersensitivity reaction. I have already made the video on type 3 hypersensitivity. So we can say that, guys, this glomerulonephritis is a type 3 type of glomerulonephritis, type 3 type of hypersensitivity. So whenever the immune complex deposited in the glomerulus, it will mediate type 3 hypersensitivity reaction. So what is the type 3 hypersensitivity reaction? So before seeing this video, kindly watch that video about the type 3 hypersensitivity, right? In the type 3 hypersensitivity, because of immune complex deposition, there is a recruitment of leukocyte. Why? Because leukocyte will ultimately remove the immune complex. It will do the function of phagocytosis. That's why whenever immune complex deposited, more and more leukocyte will get recruited. And this leukocyte can activated by Fc receptor, that is known by the name Fc receptor mediated leukocyte activation. And once the leukocyte activated, it will release certain hydrolytic enzyme which can lead to tissue injury and inflammation, and so it can lead to development of glomerulonephritis, right? And second important thing is that whenever the immune complex deposited, it will activate a complement system, right? It can activate complement system, and this complement system, whenever get activated, it will form a C5 to C9 membrane attack complex which can damage the cell membrane, right? So complement activation and the leukocyte activation is the basis for the development of glomerulonephritis by immune complex mediated. Now, as we have discussed, the antigen could be endogenous or exogenous. Endogenous means the antibody will form against the DNA, like that of endogenous cell. The classical example is systemic lupus erythematosus, while exogenous antigens could be bacteria like that of streptococci, viruses like that of hepatitis B and C virus, spirochetal antigen like that of Treponema pallidum, which lead to syphilis formation, right? Plasmodia falciparum, which lead to malaria development, and against the tumor antigen. These all can act as a exogenous antigen against which antibody can form. All right. Now, friends, you might have questioned that where the immune complex deposited in the glomerulus, which are the deposit site of immune complex. So it can be checked by electron microscopy only, right? To visualize the site of immune complex deposition, you need to do electron microscopy. So which are the site? The first site is mesangial deposit. Here the deposit could be in the mesangium. These are the mesangial cells, right? All right. Second one is subepithelial deposit, means these are the visceral epithelial cell below which you have the deposit. Below visceral epithelial cell, if deposit is present, it is known by the name subepithelial deposit, which on electron microscopy gives electron dense appearance, which is also known by the name lumpy bumpy appearance. So subepithelial deposit can also occur. Third deposit is subendothelial, means here below glomerular capillary endothelium, below endothelium there could be deposition here, which is known by the name subendothelial deposition. And fourth one is if deposit occur in this area, this is the glomerular basement membrane. So glomerular basement membrane deposit can also occur. So there are four type of site of deposition, four site of deposition of immune complex: mesangial, subepithelial, subendothelial, or glomerular basement membrane. All right. Now, whenever the immune complex is deposited, it will activate the complement. So, the immunoglobulin and the complement both will get deposited in the glomerulus, which can be seen by immunofluorescence microscopy. And it will give a granular microscopic appearance. The fluorescence will be granular in appearance, particularly in the subepithelial lumpy bumpy deposit. So, whenever the immune complex deposition mechanism operate, then it will give a granular immunofluorescence appearance. All right. Now you might have questioned that why is it so that certain deposits are in subepithelial, certain in endothelium, certain in GBM, certain in mesangial. So which are the factors that determine the immune complex localization? The main factor responsible for the immune complex localization is molecular charge. Suppose, as we have discussed, this glomerular basement membrane is having the negative charge. So if the molecule is cation, right, the basement membrane is negative charge. So if the molecule is cationic molecule, then it is of opposite side, right, it is of opposite charge, it is a positive charge. Cationic means positively charged. So positively charged molecule can cross the GBM, and so the deposit will be in the subepithelial region. Understand? If the charge is positive, right, then it can cross this glomerular basement membrane, and so deposit is in the subepithelial region. But suppose if the molecule is negatively charged, right, so it cannot cross the glomerular basement membrane. So obviously you can understand that it will get deposited inner side of GBM, that is subendothelial deposit. And if the molecule is of neutral charge, then it will accumulate in mesangium. Second factor that determine the immune complex localization is molecular size. The large size immune complex usually removed by process of phagocytosis. But if it is of medium size, small size, or slightly antigen excess, then it cannot be removed and it will localize in the glomerulus, which lead to immune complex localization. Right? So molecular size and molecular charge are the important factor to determine the immune complex localization. Now we will see the anti-GBM glomerular injury. Here the antibody will form against the glomerular basement membrane, particularly type 4 collagen of glomerular basement membrane. The antibody will form against this GBM. And this type 4 collagen, like that of structure, also present in the lung alveoli basement membrane, right? In the lung alveolar basement membrane also there is a type 4 collagen. So the form antibody will react against this GBM and lung alveolar basement membrane. So it will damage the glomerulus as well as lung alveoli, right? So there is a kidney and lung inflammation present in the anti-GBM mediated injury. So because of that, the patient will present with kidney symptoms like that of hypertension, hematuria, as well as the blood in the sputum, which is known by the name hemoptysis. This particular lung and kidney injury by the anti-GBM antibody formation is known by the name good pasture syndrome, right? It is known by the name good pasture syndrome. So it is an example of in situ antibody formation. The antibody will form within the glomerulus. And in the anti-GBM glomerular injury, remember friends, the deposition is linear variety, not granular. If you see it in immunofluorescence microscopy, then deposition is linear. While in immune complex, it is granular. Here it is linear. Alright. Now cell-mediated glomerular injury. So antibody-mediated glomerular injury is over. Now when there could be possibility of cell-mediated injury, so it is seen where the immunoglobulins are deficient, means the immunoglobulin are less, it is deficient, right? At that time cell-mediated injury will work. In this type of injury, T lymphocyte recruit the leukocyte macrophage like that of cell. It will releases the growth factor and promote the fibrogenesis. And it could be antibody dependent cellular cytotoxicity as well, right? The T lymphocyte can activate the B lymphocyte and it will activate antibody dependent cell cytotoxicity. So in which condition the immunoglobulins are deficient. This particular mechanism operate in the type 3 variety of rapidly progressive glomerulonephritis, which we will see in my next lecture, right? So it is known by the name POSI-immune rapidly progressive glomerulonephritis. POSI-immune means lack of immunoglobulin, means immunoglobulin is deficit. That's why it is known by the name POSI-immune RPGN. So in this variety, cell-mediated injury will work. Now third variety is alternate complement system mediated injury. Here what happened, friends? Here there is a persistent activation of complement system by an alternate pathway that will lead to glomerular injury. We know that complement system could be of two variety. One is classical. Complement activation can occur by two pathway, classical and alternate, right? In the classical pathway, initially there is activation of complement 1, 4 and 2, right? These four complements are activated and that will ultimately activate C3 complement. And finally by activation of C3, there is a formation of C5 to C9 membrane attack complex. So this is the classical pathway. But here what happened in the alternate pathway, this C1, 4 and 2 will not get activated and there is a directly activation of complement C3. That's why it is known by the name alternate pathway of complement activation. So in the alternate pathway directly C3 is activated and this C3 is a key complement component which lead to complement mediated injury. Understand, this C3 breaks by one enzyme, that is C3 convertase. It will stop the C3 activation, this C3 convertase. So if you have the antibody against the C3 convertase, then alternate system will remain activated, it will go unchecked, and alternate complement pathway will permanently activated. So it can lead to type 2 membranoproliferative glomerulonephritis. The example is type 2 MPGN, which is an important MCQ. Understand? Alright, now last mechanism is secondary pathogenetic mechanism. Here friends, the T cell mediated immune reaction will activate certain cells to produce the certain inflammatory mediators. For example, platelets, macrophage, messenger cells, T lymphocyte, natural killer cell will produce oxidants, cytokines, chemokines, protease, growth factor, right? Eicosanoids like that of prostaglandins and leukotrienes, then nitric oxide. So all these inflammatory mediator will mediate a glomerular injury. So that is the secondary pathogenetic mechanism. Now this is all about the immune mediated mechanism of glomerular injury. Now which are the non-immune mechanism of glomerular injury? So here the examples are metabolic cause like that of diabetic nephropathy, hemodynamic cause like that of hypertension induced kidney injury, then amyloidosis, right? Then infections like that of hepatitis B and C virus, E. coli, HIV virus, etc. Then certain drugs, NSAIDs like that of drugs, and Alport syndrome. These all are the example of non-immune mechanism of glomerular injury where the immune mechanism is not involved. All right. So this is the final table from the Harsmohan book which shows the different mechanism operate in a related glomerular disease. If we see it in some more detail, then basically it could be immunological mechanism or non-immunological mechanism. Non-immunological mechanisms are hemodynamic, metabolic, right, infection, drug, etc. that we have already seen. Now in the immunological mechanism, as we have seen, it could be of 3 to 4 variety. One is antibody mediated, then anti-GBM disease, then cell mediated, and secondary pathogenetic mechanism. The example of antibody mediated injury is by the immune complex. Main mechanism is immune complex mediated injury. The examples are diffuse proliferative glomerulonephritis, membranoproliferative, IgA nephropathy, membranous nephritis, like that of, understand. And the example of anti-GBM is good pasture. Alternate pathway system operate in membranoproliferative type 2, right? And cell mediated mechanism operate in the POSI-immune type 3 variety of RPGN. We will see the each mechanism in the detail in individual type of nephrotic syndrome. So this is all about the pathogenesis of glomerular injury, various pathogenesis of glomerular injury. To sum up with the summary, the glomerular injury could be immune mediated or it could be non-immune mediated, right? The example of immune mediated is by the immune complex deposition, by the anti-glomerular basement membrane antibody formation, right? Cell mediated, alternate system activation, etc. While non-immune includes diabetes, hypertension, infection, drugs, etc. So this is all about the pathogenesis of glomerular injury. Hope it will make your fundamental clear to understand the further lectures of nephritic and nephrotic syndrome. Thank you very much. Take care and see you soon in the next video. Till then, take care and bye bye.